Hello students, welcome you all on my channel. In this video, I'm going to discuss about the excretory system of mammals. In this video, I will discuss about the position, structure and blood supply of cortical and juxtamedullary nephron. Here, with the help of a realistic diagram, I will demonstrate about the cortical and juxtamedullary nephrons location and the function. So without wasting time, I'm going to draw the diagram of the cortical nephron first. This is the cortical nephrons. This is descending going. It is the loop of Henle I'm making. To make a difference, I'm just making the thin segment. This is the thin segment. And now ascending the thick segment. And now this is the, as you know, this is structure. This structure is known as the blind sac Bowman's capsule. This is structure. This is the blind cup-like structure. This structure is PCT, just located near the Bowman's capsule. I will make the glomerulus filter here. This is the thick segment of ascending loop. This is the thin segment of ascending loop. And this is the DCT. Now it is joining with. It is joining with the collecting tubule running down and forming pelvis in the kidney region. This is the collecting tubule. There is many branchings are there of the collecting tubule. It's a very easy diagram. You can make it, and now you will understand the location of the kidney, location of the nephrons in the kidney. This region is called pelvis. This is the collecting duct, and this is the branchings. Now one of the here with the help of this one, this is the I'm going to make. This is the structure of the juxtamedullary nephron. Juxtamedullary nephron, its loop of Henle running down in the medullary region. Now I'm going to make a thin segment with the help of another sketch so that you can understand. Now again the thick segment starts. this structure is a juxtamedullary nephron, and this structure is the cortical nephron. Just it will be clear when I will make the blood vessel here. This blood vessel is a renal artery. Below it, there will be renal vein. Now I am going to draw the structure of efferent arteriole, from where the efferent arteriole is coming and where the efferent arteriole is going, and what the role of efferent arteriole and what is the role of afferent arteriole. That will be clear when the diagram will be seen, or when you will be focused here. See, this structure is a renal artery, and this one is the renal vein. Renal vein. So, as you know, the artery breaks into the fine arterioles. So here, a structure arising. This is the arteriole, and this is breaking into finer structure and forming globulari in the Bowman's capsule. Now, from the Bowman's capsule, this structure is coming. This structure is known as efferent arteriole, and as you know, it is a running parallel to the loop of Henle. It is running parallel to the loop of Henle and mixing into the Henle venule. This may be arising again, some other blood vessels will arise. So this is afferent arteriole. Afferent arteriole. This one is efferent arteriole. This structure is called Bowman's capsule. This structure, this convoluted structure, this is known as PCT. PCT means proximal convoluted tubule, and this structure, this is the convoluted, but it is far from the Bowman's capsule. This structure is convoluted but far from the Bowman's capsule. So this is DCT. This is the DCT. This is the loop of Henle. This is the loop of Henle. The loop of Henle. It has two parts, ascending and descending. So this is the thick segment of ascending loop of Henle. Loop of Henle. This is the thin segment. Thin segment of descending loop of Henle. And this structure, this is called, this running parallel in the juxtamedullary nephron, the capillaries are running parallel to the loop of Henle is termed as you know, it is termed as vasa recti. This is this capillaries collectively it is termed as vasa recta or recti. Now come to this portion. This is the cortical nephron. Again the same. Here will be glomeruli, and this glomerulus is coming from this area, from the renal artery. Arterioles are emerging, and then it is going into the Bowman's capsule, and then it forming network here. This is the afferent arterioles or artery. Now it is coming again, zigzag like structures surrounds the entire nephron, and then mixing here. Now I had told you the capillaries are running parallel with the juxtamedullary loop of Henle is termed as a vasa recti or vasa recta. But here the capillaries which surround the rest of nephron is known as peritubular capillaries. This structure is called peritubular network of capillaries. Now the point is come to divide the regions here. So this region here I am making structure. This region is cortex part. Always remember, see, the cortical nephron lying in the cortex region. That's why I have divided this with the help of dots. And the remaining portion is the portion. This portion is medulla part of the kidney. See, students, here I told you earlier that the cortex part of the kidney is rough and darker in appearance because of presence of Malpighian corpuscles and PCT and DCT. Same here in juxtamedullary nephron also. Juxtamedullary nephron lying in both the region, but the cortical nephron lies only in the cortex region. That's why it gives a rough appearance to the cortex part of the kidney. Now here come to the juxtamedullary nephron. In juxtamedullary nephron, when you see this structure, the loop of Henle lying in the medulla part of the kidney. That's why the medulla region of the kidney is lighter and striated in appearance because of the larger loop of Henle. And here the Bowman's capsule, glomerulus, PCT and DCT, they are present in the cortex region. Because of the juxtamedullary nephrons, PCT, DCT and the Malpighian corpuscles, and the cortical nephrons, PCT, DCT and the Malpighian corpuscle, they are rough in appearance and darker and granular in structure. Now come to the main point here. See how glomerulus is forming. Just I'm explaining you here. Afferent arteriole, focus here student, afferent arteriole bring blood to the Bowman's capsule. There it breaks into fine capillaries, and these capillaries form network of glomerulus filter. After filtration of blood is drained out by efferent arteriole. Drained out by efferent arteriole. In the case of cortical nephron, the efferent arteriole form a network of capillaries which surrounds the rest of the nephron and form peritubular capillaries. And later these peritubular capillaries join together and form a renal venule, and these renal venule again join with the renal vein. So it is clear about the cortical nephron. Now come to the juxtamedullary nephron. The same thing is happening here. The efferent arteriole bring blood to the Bowman's capsule. There it breaks into the fine capillary network, form the glomerulus, and in the glomerulus, in the Bowman's capsule, the filtration of blood is taking place under high pressure, and the filter blood is again taken by the efferent arteriole. This arteriole, efferent arteriole breaks into the fine capillaries and run parallel to the loop of Henle, forming vasa recti. A vasa recti in the case of juxtamedullary nephron. And then it joins with the renal vein. See, students, the descending loop, the thick segment of ascending loop is made up of a squamous epithelium and impermeable to water. But the thin segment is made up of cuboidal epithelium and it is permeable to water. Here I want to tell you about one hormone which is secreted from the afferent arteriole's epithelial cell, and this is known as renin hormone. This renin hormone helps in the reabsorption of sodium ion when the sodium demand is maximum in the body. So renin angiotensin mechanism play vital role in the retention of the sodium in the body. That I will discuss in the later when I will especially take the topic of the renin angiotensin mechanism, and then I will explain you. See, another hormone which is secreted from the peritubular capillaries of the kidney, and this hormone is secreted from the cortical region of the kidney because the peritubular capillaries aid in secretion of the hormone. The name of hormone is erythropoietin hormone. This hormone aids in formation of blood corpuscle by enhancing the stem or RBCs. RBCs is forming from the stem region of the bone marrow. So it activates the region and aid in the formation of the blood. Now come to this point about the collecting duct. Here the urine is taking place, means urine is formed and now urine is moving to the pelvis region. Now as I told you in my previous video that the Malpighian tubule, this glomerulus and the Bowman's capsule play a crucial role in mechanically filtration of the blood. PCT reabsorbs 75% of water, glucose, salts, amino acids, etc. DCT act as tubular secretion, play a vital role in the secretion of H ions, certain drugs, potassium, etc. Loop of Henle act as countercurrent and maintains the concentration gradient of urine. That is to say, you may see that's why in certain mammals like kangaroo rat, the urine is almost semi-solid because of the larger loop of fennel. And why it is semi-solid? Because kangaroo rat never drink water. It uses metabolic water. That's why it has to conserve water. So that's why the loop of Henle play crucial role in the reabsorption of water there, and play which maintain the concentration of the urine. But in the case of human, maximum work is done by the cortical nephron, because this juxtamedullary nephron play crucial role in deficiency of water supply. But when the water supply is maximum, when the water is present in the body, then obviously this task will be taken by the cortical nephron. I have forgotten to mention this one. Cortical nephron, and this one is the juxtamedullary nephron. Students, this structure, this large nephron is known as juxtamedullary nephron, and that is known as cortical nephron. This structures, the small structure coming out from the collecting duct is called the branch of collecting, branches of collecting duct. The kidney has a structure, the external structure, that structure is called the fibrous capsule. Fibrous capsule of kidney. It is a protecting covering, and as you know, I have divided the region into cortex and medulla. It is clear. And please, students, please make the diagram. Now, students, there is a more remaining portion of the excretory system is here, but because of the video is long, I have to split the segments. So in my next video, I will discuss about the ureters, urinary bladder, urethra, and physiology of excretion. That will include ornithine cycle and urine formation. And if there will be possibility, then I will complete the diseases of the kidney. Thank you very much for watching my channel and this video. There is a lot to come. Thank you very much.